Okay, dear students, we were talking in the previous class about archibacteria. So let us continue with the same thing here. So what are archibacteria? Archibacteria are nothing but they are the prokaryotic organisms with the single cell organisms which lack the nucleus. So they also lack the peptidoglycan in the cell walls and have the different lipids in their cell membranes. So membranes, they have the lipids. Then different types of ribosomes are also seen in the archibacteria. Very different gene sequencing. So gene sequence is also different from the other bacteria in archibacteria. Why? Because they are present in the very extremely harsh and the unsuitable or unfavorable conditions they are found, and they are anaerobic, due to which they have the different gene sequencing than the normal bacteria. Okay. Now, bacteria can live in extremely harsh environments, and they do not require oxygen and can live in extremely salty environments as well as the extremely hot environments. So, as it's a salty environment and hot environment they exist, so the other organisms cannot live in such conditions. That's why these are different from the other microorganisms. So the archibacteria are also called as ancient bacteria. Next, archibacteria are subdivided into three groups. Those are nothing but methanogens, thermoacidophils, and extreme halophils. So these are the three groups where the archibacteria are divided. So we'll see one by one now. Coming to the methanogens. Methanogens, these are the organisms which live in anaerobic environment. Means in the absence of oxygen, they can live, okay, or they can survive, and they get the energy by changing hydrogen and carbon dioxide so into the methane gas. So what they do, whatever the hydrogen and the carbon dioxide which is present in their environment, these microorganisms or the archibacteria, methanogen archibacteria, they convert this hydrogen and carbon dioxide into methane gas. And we can see these methanogens in the swamps and sewage treatment plants, and also in the digestive tracts of the animals. So by the smell, we can make out in the animals when they pass, we get some smell that is nothing but a methane. Then methanogens, they break down cellulose in a cow's stomach. So the cellulose which are present, the plants have the cellulose, and what are the plant material these grazing animals they have, that is broken down by these bacteria. So these bacteria are very important for the grazing animals. So also they produce marsh methane gas. So by the digestion of the cellulose, they convert that cellulose into the methane gases by the use of the carbon dioxide and hydrogen which is present. Then next one is halophils, that is extreme halophils. Halophils means salt loving archibacteria. So live in very salty water, and they use salt to generate ATP. So adenosine triphosphate, that is nothing but a energy. In PUC and all, you must be knowing those things, these terms. Okay, then the third one is thermoacidophils or thermophils. They are also called as thermophils. Thermophils mean, thermo means related to the temperature. That means hot, extreme hot environments. So these bacteria lives in extremely hot environments. They are found in volcanic vents where the volcanic eruptions and all take place. There you can find these archibacteria, where we all cannot go, or the animals cannot live in such hot conditions or the hot environments. So whatever the, if plants or any other microorganisms, they will go, or they will be found in that place, they will die. So but these archibacteria, they are very they love this such type of environment, and they grow there very nicely. So hot springs and cracks on the ocean floors that leak acid, so in such conditions they can live. So then coming to the reproduction of the bacteria. So bacteria reproduce by asexually by binary fission, where sexual what we call sexual mating will not takes place by the division of the single cell into two parts or more than two parts, and those parts they grow into an individual similar to the original ones. We call such type of reproduction we call it as binary fission. And these bacteria have single chromosomes, replicates, and then cells divides here. They are rapid, means very fast. All new cells are identical, or we also call these identical cells after binary fission, we call them as clones. Okay, clones are nothing but they are an exact copy of the mother organism. They are the exact copy. It may be a virus or bacteria, and they are scientifically they are produced. Scientifically means like binary fission, scientifically breaking those into different parts, one or two or more, and they all are in same size, shape, and having the same genetic material. Means identical they are. Means exactly the same copies of the mother. Mother, or it may be a parent. We call it as mother or parent. Okay. So the binary fission in bacteria. So this is a bacterial chromosome, and this outer covering is the plasma membrane. Next to that is a total out of that plasma membrane you can see a cell wall. Then duplication of the chromosomes takes place, means doubling of the chromosomes, and it continues to grow, and the cells then divide into two. Then division of the two cells, and these two cells are exactly the exact copies of the original cell or the parent cell. Then binary fission in E. coli. This is a figure of E. coli, Escherichia coli. This is the example for the bacteria. Bacteria reproduce asexually by conjugation. Conjugation is nothing but there is a tube-like structure developed between the two cells. Form a tube between two bacteria to exchange genetic material. So the two bacteria they come together, and they uh, they exchange their genetic material held together by the pili. So pili you have seen that is a um, the it is smaller than the flagella, thread-like structures which are found on the cell wall which um, means inside the cell wall these pilias are present. They originate from the plasma membrane to the outer side. We call them it as pilia. They are thread-like structures, and they are proteinaceous, and new cells they are not identical. So whatever the here the conjugation or the reproduction which takes place by conjugation method, the new cells are not identical, whereas in binary fission they were identical, but in conjugation method the two cells are or the new cells are not identical. See here, so one is bigger and another is smaller. So these two bacteria they come together and they form a tube like structure. They connect with each other, and they exchange their genetic material from one cell to the other cell. So their size also varies here. One is big and one is small. Sorry. Then after that we can see in this spore formation. So for these bacteria they form endospore. Whenever habitat conditions become harsh. So habitat conditions become harsh means adverse conditions, means environment is not suitable for the bacteria to grow. So that time these bacteria what they do, they undergo um, uh, in the formation of spore, that is endospore forming. Further they are not going to perform any activities. Means in dormant period they will form a dormant period. Means they will sleep for a while until the favorable conditions comes. So sleeping mode they go, and until the favorable condition comes. So that we call it as harsh situation for them, and they able to survive for longer periods of time as endosperms. So until unless and until they get a favorable condition, they go for a long sleep. Means they can survive, means they are alive, but in dormant period they will be. Means they will be in sleeping mode. As soon as they get the favorable condition, they again start their activities. So difficult to destroy these spores. So bacteria we cannot destroy easily, and they are heat resistant. Heat resistant means they can tolerate as much of heat. Means high heat tolerability they have. So they can tolerate very high temperature also, hot conditions also they can survive. So we cannot destroy these bacteria easily. Then coming to the transduction and transformation in the reproduction. So transduction is nothing but it's the process by which uh, foreign DNA is introduced into a cell. So transduction means nothing but introducing a foreign DNA. Means some other DNA is introduced into a cell of one bacteria by a virus. So here the genetic material is transferred by a virus into the bacteria, or the viral vector also we call. Means virus acts as a carrier or a transformer here, which carries a genetic material and it transfers. Okay, then transformation is nothing but a genetic alternation of a cell. Means genetic alternation means after transformation there is a alteration. Means if you add something, then if before we have some genetic part, and to that new genetic material if you are going to add, then obviously there is a change in that genetic material. So there is a genetic alternation of a cell, which results directly uptaking or incorporating exogenous genetic material. Means uptaking means it may uptake means it may take inside, or incorporate means introducing the new genetic material into the bacterial cell from the surrounding area or the surrounding environment. Through which? Through cell membrane. So cell membrane, through cell membrane you are going to introduce what? Other genetic material directly, or incorporating it through exogenous genetic material from the surrounding. From the surrounding you are going to transfer those genetic material into the cell. So that is what we call transformation. So genetically change the bacteria here, and may become antibiotic resistance. So when we give antibiotics, so sometimes they don't work, so they, because they become resistant to that antibiotic. Okay, then transformed bacteria pick up the pieces of the DNA from the dead bacterial cells, and transduction viruses carry foreign DNA to a bacteria. So what I told, so viruses they act as a carrier or the transporter, which add the foreign DNA into the bacteria. So this is all about your bacteria. Dear students, if you have any doubt, you can ask by calling me. I hope you have my number also in WhatsApp group. Thank you, students.